All right, thanks very much, everyone. Thanks for bearing with me, especially if you were expecting to see me last week. I really wasn't in a fit state to do anything last week, so thanks very much for bearing with me. So I'm going to be talking about a seascape approach to the protection of essential fish habitat. Hopefully. So I'm going to set out my stall right from the start. So I think the seascape approach that I'll be talking about today is an extremely useful way of thinking about how we use the sea and things like that. For nursery areas, I think it's really important that we need to consider not just the types of seabed substrata, but also the mixtures of patches. And the scale I'm thinking about here is really like at the scale that's relevant to the sizes and movement patterns of the juvenile fish. I think looking at seascapes is also really useful as a way of thinking about how to protect marine biodiversity, particularly as we're going into the question of new highly protected marine areas. And I think thinking about seascapes is also really useful when we're thinking about restoration and monitoring, especially thinking about where to prioritize in terms of where should be restored and the sizes of the areas and that sort of thing. Okay. So what we're going to do today then, I'm going to talk a little bit very briefly at the start about the people who actually did everything, because the vast majority of it was not me. How we did this work, what we found out about juvenile habitats, and then moving into the ideas of seascape ecology and how we came to those. And then one of the big issues, obviously, is thinking about bottlenecks. So are there stages in life histories or particular habitats that cause bottlenecks? And so you could have plenty of juveniles going in, but not very many coming out. And then some suggestions about, well, where we're currently going with this work and other things which are worth doing afterwards. So who did all the work then? So I'm going to be talking about these people as I go. These are the real kind of heroes of the story, mostly PhD students. And they're kind of in a sort of historical order, in basically the same order that I'm going to be using for the purposes of the talk. Start with Mary Ryan, who wrote our first work on nearshore fish in Scotland, through to Natasha, who's currently the lead on our work in Arran. And I'd like to call out the colleagues from Nature Scot and Marine Scotland who have been our partners with basically all of this work. Okay, so just a little bit about who I am. So I started my career working on tropical marine resources down in York, mostly in Egypt. And then so groupers were my speciality there, the sort of medium-sized predatory fish down in the photo on the bottom right. I then went off to Antarctica for my PhD work. If you can see my cursor, that poor sucker there is me carrying the equipment for Katrin, who is the actual expert from Germany. So I did my PhD work in Rothera station down in Antarctica. So I went from working on the physiology of Antarctic animals into deep sea animal physiology. If you've heard of me, that's probably if you've heard of me, then that'd probably be why. So I ended up as scientific PI for the big observatory program in Angola. So putting these enormous observatories in the water off Angola to monitor the deep ocean environment. Done a little bit of work with AutoSub, and but the mainstay of my work was using things like this camera system on the right. This is one we called Shocker, and its job was to give electric shocks to deep sea fish so we could see how fast they can swim. And it was an enormous, great camera system, about 3 meters high. And we deployed that down to anything down to like 5,000 meters. But I I did quite a lot of work with beta cameras, and I got really interested in beta cameras and how you understand the world from beta cameras. So how do we turn the information we get from beta cameras into useful information? And that turns out to be a much more complicated thing than I expected. And now I've kind of moved on to looking at temperate zone fish, and this has ended up being a very large part of my work. The group as a whole we've written papers. This stuff just since I've been at Glasgow. Each dot is a place where we've published work from. We have other places where there are ongoing projects, but these are the ones where papers have been published from already. So a kind of scattering of deep sea work in the Pacific and the Atlantic, quite a lot of tropical work from the Caribbean through to Bangladesh, from coral reefs, coral reef fish, and mangroves and mangrove fisheries resources, and right the way down to still another paper from Antarctica, which I'll talk about a little bit in a second. So it kind of looks like it's kind of all over the place. I kind of sometimes feel kind of like this guy, the it's all connected meme guy, trying to explain to people what all this is about and what actually joins it all together. And this is a word cloud I did from the titles of the papers that we've published since the group formed in Glasgow. It's overwhelmingly work on fish. So we have a few papers and other things, quite a lot of deep sea, a lot in the Atlantic, but really lots of different things, but basically revolving around the lives of fish. So as Hannah said, I did my last postdoc over in California at Scripps, and I came back to Scotland to take up a lectureship at Glasgow. And quite early on, I was contacted by Nick Bailey and Bill Turrell to join what was called at the time the West of Scotland Task Force, which was a group of scientists from academia and from Marine Scotland and fishermen who are interested in trying to understand the state of the West of Scotland, because at that point it was already recognised that there was a bigger problem on the west of Scotland than there was kind of in general in Scotland. So we got together to do a kind of review of the state of the marine environment on the west of Scotland. And I was given a kind of free hand to whatever I wanted to look at for this, but it kind of occurred to me that it would be interesting to look at the fish, the close to shore fish, the nursery areas and juvenile fish and that sort of thing. And this is where luck really kind of came to me, because it turns out that Mary Ryan had just arrived in the UK and was looking for something to do, just happened to be an expert on juvenile fish from Canada. So you've been involved in a lot of the work on kind of seagrass and fish interactions there. So we wrote a chapter together, or led by her, but with me, on nearshore fish in Scotland. And I think we were both surprised about how little was known, even though it came under the responsibilities of quite a few different people. There are a huge number of unknowns about juvenile fish in Scotland, even compared to how difficult the stuff is to study in general. Knew very, very little about what was going on in Scotland. That led then to a Scottish Industry Science Partnership project to develop ways of studying inshore fish. And one of the things I was very keen to do then was to bring in the use of beta cameras, because I knew how kind of effective they were in the deep sea, and I thought they might be useful for the work that we had in mind. So like I said, I've had a long interest in the use of beta cameras. I think they're extremely useful approach is that this is a paper we wrote called Cameras and Carcasses, about the use of cameras in the deep sea. But most of the things here are still true of wherever you use them. So this looks at the pros and cons of the different approaches. And one of the main things which beta cameras are really good for is that you can get a lot of data relatively quickly with quite simple infrastructure, without huge amounts of training of the staff necessary to get the stuff in the water. And tend to be kind of non-destructive. They don't damage the seabed or other kind of infrastructure on the seabed. But one of the issues is they tend to be, the way they're analyzed is not standardized. It's very difficult to get absolute abundance estimates from beta cameras. And there are a lot of assumptions about the evidence that you get from them. Basically, I think you need a lot of beta camera data in order to be able to say anything useful, and there are a lot of caveats to it. But we'll get into that later on if you like. So this is our first go at a beta camera. This is Kathy Dunlop. So she was the first PhD student to work on this topic, and this is the camera system that we built, which was used in the UK, mostly in Firth of Clyde and the west coast of Scotland, but also a similar version which we used in Eilat in the Red Sea as well. And then this is her deploying it in Antarctica. So the different parts of the frame are labeled, but the camera is basically a time-lapse stills camera. And there's acoustic current meter in the center of the unit, and then the lights were off an angle, held off to the sides, and in bait in front of the camera. And this was extremely robust and kind of effective system for getting abundant or basic information about the arrival rates and numbers of different species. And Kathy used this to validate different methods of assessing abundance from beta cameras. And then from there, we moved on to what are called BRUVs, baited remote underwater video systems. So these, each one of these systems has two video cameras in it. So the one on the left is the older model. So this is what has been used for all the work around Arran. So two video cameras in the white and blue housings with a current meter in the middle, and then the bait in the white box. And then on the right is the newer models with GoPros in, which are there being used in Loch Erebor, which is the new study site, which was studied for the first time last year, and which will form hopefully part of a wider program of this work. So the advantage of stereo video is you can take measurements of animals, you can quantify the size of the field of view, but you can also measure the sizes of the animals. So therefore you're able to start getting things like biomass estimates. Yeah. So they're extremely effective, and they're quite small and easy to deploy, especially the new ones. The GoPro ones are really effective and easy to deploy. And you can get, I think Neil and Charlotte, when they were working up here, they were getting up to about 15 deployments a day out of 3 systems. So you can really crank through the work with these. So what we're mostly going to be talking about today is this idea about essential fish habitat and using this information to understand what it is. And this is from the US Magnuson-Stevens Act, so the long established and generally regarded to be very effective bit of fisheries legislation. So the US has been ahead of us in terms of the recovery of fish stocks for a while, and the protection of essential fish habitat has been a core part of the necessity of their fisheries management plans since this act came in, because they recognise, say, one of the greatest long-term threats to fisheries then is the loss of habitat. And they have this really unique kind of hierarchy where they think about the most basic level. So level 1, the most basic level is just presence absence data. So where do you see fish and where don't you see them? And there are loads of information on that. It's relatively easy to get. So the bars on the left represent basically how much information is available. So level 2 would be having both presence absence and some measure of population density. Level 3 also adding in information about growth and survival. And then finally, the highest level that they have the lowest amount of information on so far would take you right up to being able to work out what parts of the environment have the greatest effect on adding fish into the wider population, sort of the most productive habitats. And that's really the gold standard. It's where we really want to be able to get to, is to be able to say, what parts of the seafloor have the greatest effect on the state of a population, usually thinking about fisheries, but also it could be the population of something that's protected that we want to rebuild a population of. So different scales that we need to consider when we're doing this. So there's the point scale. So when we see a fish, we can look at the immediate area around it, and that would be the point scale. So this would be the classic kind of cod seagrass interaction that gets talked about quite a lot. So we can say at that point where you see the fish, what kind of seabed is that? Okay, so that'd be the point scale. And then you have a higher scale, you have the region going right the way up to the whole range of the species, which is where whether the fish is present or its abundance then is driven by larger scale things like the light level, sea surface temperature, food availability, that sort of thing. Food levels as driven by primary productivity, say. So we tend to know a reasonable amount about these things. But there are two scales going in between. So there's the patch scale. So what do we know about the shapes and sizes of patches of habitat of different sorts? If we see this fish in this piece of seabed, is that the only bit there, or is that part of an enormous expanse of it? Is it part of a whole load of different patches? And these make up the seascape. Well, what patches? How big are the patches of the habitats that we're interested in, or the seabed types rather that we're interested in? And do they form a network of patches with other sorts of patches in between? What are those mixtures? What are the boundaries? That sort of thing. Are there barriers across it? And we tend to know quite a lot about the point stuff and the region stuff, and much, much less about the patch and seascape stuff in between. So we've done some work on considering the kind of regional stuff with whiting. So this is work that was done by Neil Burns and supervised by Peter Wright, as well as ourselves up in, Peter Wright from Marine Scotland Science. So this is stuff that's done using Otolith microchemistry. I could spend the rest of the talk explaining how this all works, or at least trying to, but I'll explain what the end result of it was at least. So these diamonds in panel K are the capture locations of adult whiting. Then if you go to the individual panels, say panel B, so this is where an individual whiting was caught, and the colors indicate where it was most likely that that animal lived when it was a juvenile. So you can see that for most of the diamonds, the most likely place that they were juveniles was in the Firth of Clyde, Solway Firth, that sort of area. So we're able to say to some extent on a kind of regional scale where are the most important areas for whiting nurseries. So we know that it's something about these areas that makes them important as whiting nurseries, but we don't know specifically what. And we can make some assumptions based on what we know about the kind of general seabed, but we probably wouldn't want to be closing these entire areas to fishing based on protecting of these nursery areas. They might want to have some special measures in there, but it would be important to work out within these where the most important areas within this were. So this brings us on to our detailed study site. So we've done most of our studies around the Isle of Arran, which is a large island in the Firth of Clyde. I'll show you exactly where it is on the map in a second. So our first kind of campaign on this was led by Sophie Elliott. So that's who you can see in the top right there with the camera system. This is 2 summers of work. In the first year, we did a mixture of diver operated video and BRUVs. So 31 diver operated stereo video transects, 100 meters long each, counting the fish and working out what the dominant substratum type was. 260 BRUV drops, and then for each one, max N, so the maximum number of fish seen in the field of view at any one time, and then what the dominant substratum was, so the substratum that covered the most of the frame. The diving was great fun, but the study site goes down to more than 30 meters, and doing 3 dry suit dives a day to that depth with that very unwieldy camera system proved to be pretty exhausting, and obviously difficult to get a suitable number of properly qualified divers together and things like that. And it turned out to be not really workable for the 3 data points a day that we were getting compared to how productive the BRUV work was. So these are the locations of the BRUV drops then. So the panel on the right, you can see where the study site is in Scotland. So it's kind of southwest of Glasgow in the Firth of Clyde. It's a very large, relatively large island. And then the study site is in the South Arran marine protected area, which stretches all the way around the south of the island. And the dots are the BRUV drop locations, and they're coloured by the dominant substratum that was seen there. You can see kind of mud further out going into sort of gravel, and then into kelp-dominated boulders close to the shore, called boulder and rock close to shore. There is a very small amount of seagrass here, but it is never proven to be particularly kind of interesting in this area in terms of numbers of fish compared to the other seabed types. So we're going to start with the data on sort of point results. So you look at the figures on the right hand side. We have cod, haddock and whiting, and the results here are the square root of max N, so max N being the highest number of fish seen at one time. And we have ABC is algal, boulder and cobble, algal, gravel, pebble, sand and seagrass. And the gravel here includes the merle as well. It wasn't in the data points we had, not enough merle on its own to make it a separate category. And the different colors are different years. So 2 kind of general things you can see. So cod in the top panel has a very different distribution of juveniles compared to the other 2 species. Even these are all gadoid fish. Very many more in the gravel, the AGP seabed type, than the others, which tend to be much more in the mud and sand. Interestingly, the cod in the top one, you can see that in the year when there were lots of cod, so if you look at the AGP bar, when that is highest, you see cod across quite a wide range of substrata. In the grey bars, when cod numbers were much lower, they're very much more concentrated around the gravel, which makes me think they basically would much prefer to be in the gravel, but when they're in higher abundance, they kind of spill out over everything else. But yeah, in general, quite a clear difference between between cod and the other 2 species. The other thing that was interesting is the effect of benthic biodiversity on max N. So for both cod and whiting, there's a positive relationship with Shannon diversity, but down for haddock. So this kind of points us to the idea that seabed quality is important for at least two out of the three species in terms of the relationship with the maximum number of fish present. So I'm going to talk about how we got then onto thinking about seascape stuff. So anyone who's talked to me about this will have heard me banging on about black grouse quite a bit, because this is how I kind of accidentally got into thinking about this. So I was working on a wind farm project as part of a community group, and the RSPB were very worried about the black grouse in the area. So this is all stuff from the RSPB that I'm showing here. So these are black grouse. They have an interesting kind of life history where they do a lot of like displaying on called leks. They're very visible then, but they need quite complicated set of environments. Their general distribution is they're in green on this map. So they're, an upland species now, mostly away from cities and in higher ground, but their requirements are like much more complicated than this. So this is what the RSPB expert told me. So they need trees for nesting, and the adults will feed in the trees. They want short grass for lekking, which is when they display of the adult males. Then long wet grass for the young ones to hide in and feed in on insects and things. And they need all of these kind of close together, right? And so it's no good just to choose the habitat where you see most of them and think, well, they like that, so we better have more and more of that. So, but on that basis, you'd say, well, we see them mostly in short grass, right? Because they're most visible when they're lekking. So we better cut all these trees down and drain all the rest of this, so they've got more of that short grass that they obviously like the best. But if we did that, they would all die, right? Because they need this complicated kind of mosaic of different sorts of environments very close together. And this is probably why black grouse are in some trouble, right? Because they have this very complicated habitat requirement, including lots of edges between things. So this is landscape ecology thinking. And terrestrial biologists have been on this for a really long time, obviously, but we're just starting to get to this here in the marine environment. There's a few groups who have done some really good work on this, but it's still quite early days for the application of this to the marine environment. So could we apply these ideas to seascapes? So what Sophie did then was to use the camera data that she had to create a seabed map for South Arran. And now we've got this has been shaded. So this is done predictive model using general linear model and ArcGIS to create these maps. So you can see the different colors then. So the brown is mud, the kind of beige color is sand, the yellow is the gravel, and the green is the kind of kelpy rock environment. And so she was able to create this map then of the seabed. And then what she was able to do then was to relate the max N of an individual point on the seabed, not just to what was actually like at that point, but to a radius of around 1,500 meters around it. So the model that she created allowed us to look at so this N infinity is a Hill number for dominance of the substratum. So in a 1,500 meter radius circle around the point, she looked to see to what extent that circle was dominated by one substratum type. And what you find then for cod, Gadus morhua, is that they were much more likely to occur occurred in greater numbers if the seabed wasn't dominated it wasn't highly dominated by one substratum. For haddock and whiting, it was the other way around. So high level of dominance by one substratum type was good as far as they were concerned. So these are our first kind of looks at kind of seascape ecology. So cod, we found, they're very strongly associated with gravel, lower abundance, where the substratum was dominated by one type within a sort of 1,500 meter radius. And that radius was chosen based on what we knew about the swimming speeds and mobilities of juvenile cod. Whiting and haddock were very different. They don't like the boulder, algal covered boulders, but apart from that, they live pretty much anywhere, and they would like it basically. The more dominated by one substratum type, the better. So you can start to see already why this would be important, and why how this might have a bearing later on in terms of bottlenecks, being generalists versus specialists and that sort of thing. So next phase then, so this is led by Natasha Walker-Milne. It's the current PhD student on this work. So she extended the time series, bringing it up to 554 bruv drops. And for each one of those, she recorded fish presence, absence, and then the percentage cover of the different substrata within the field of view of the camera. This is augmented with 333 camera drops given to us by Scottish Natural Heritage, and that was used for substratum percentage cover. And also 30 fish traps done by Marine Scotland Science, who are also co-supervising this work. And the traps are used both to look at the timing of arrival, how long fish were staying in the nursery areas for, and to look at growth rates. So we're trying to move our way through that kind of NOAA hierarchy towards understanding drivers of growth as well as presence, absence, and abundance. So with this much higher number of points now, so we're getting towards 900 images of the seafloor, Natasha was able to create a very much more detailed seabed map. This is done using krigging, or ordinary krigging, rather than a GLM. And what she's been able to do is to look at metrics of the different patches. So she's able to look at patch size, area, length to area, and all that sort of thing. This is all kind of continuing work, so I'm going to be fairly cagey about all that shows for now. But she's interested to know to what extent the characteristics of these patches drive things like benthic biodiversity. But that work's not complete yet. What we do know is what drives the likelihood of seeing our three different focal species. So for each one of these, the y-axis is the probability of the species being observed. And what we have then is the different significant drivers for cod. So they like, so starting from the left then, so they like more algae up to a point, so up to around 50–60% algae. You're more likely to see them, so tailing off towards very high coverage. You tend to prefer, or more likely to be seen shallow, sort of top 10 meters. Much more likely to be seen where there's lots of gravel, so the figure on the right, more gravel the better. Terrain ruggedness, so rougher ground. And lastly, but no means least, Simpson's diversity. So much more likely to be seen if the seabed had high Simpson's diversity. Again, pointing to this idea that habitat quality is important as well as habitat type. Haddock different then, so they don't like algae. They be seen much deeper, typically around 30 meters, low current, also high Simpson's diversity, and close to shore. So relatively deep water, but close to shore, but also this link to benthic biodiversity. And whiting, so as much mud as possible, also around 20, 30 meters. They like lots of sand. They don't want the terrain to be rugged. They don't want high currents, but they do also want high symptoms diversity. And what you get then, if you're looking at the distributions of the different species, you can see from left to left you have cod, then haddock, then whiting, basically kind of shaking out into a kind of almost like a zonation pattern around Arran, with cod also likely to be seen in a kind of patch of sand down there off what's Pladder, basically. So from the point data then, what we can say is that cod like the shallow, rough, algae-covered gravel substrata, whiting and haddock deeper, less algae, more mud and sand, and they kind of shake out into sharing that space, but all more likely to be where symptoms diversity was higher. So kind of interpretation of that, the quality of the habitat is critically important. So protecting habitat just when they're occupied is probably not enough, because you want to make sure the habitat is in good condition as possible, not just that you don't kill the fish while they're there. For cod, it's habitat protection very close to the shore. Haddock and whiting probably need to be protecting the soft sediment in the sort of shallowest 30 to 40 meters. So that's the presence absence data. But like I said, we're going to try and look at growth as well. And things get even more interesting when you get to growth. So I'm just presenting a kind of subset of the growth findings here, just for ease, really. So the data here show growth rate based on the size change of the otoliths. And for whiting, kind of unsurprisingly, you get a kind of a progressive increase in growth rate with increased proportional coverage of mud, but then dropping off at very or much higher mud levels. But still, their growth's at highest at what's still a relatively small proportional coverage of mud, surprisingly. Natasha's also done a load of work looking at boundaries of the areas, so looking at the relationship between the distance inside and outside of particular patch types. What's interesting for whiting is their abundance seems to be highest around the kind of edges of sand patches. So increasing, so looking on the right there, the dot, it will be the boundary of the patch, and then you basically have their growth rate going higher as you get closer to that boundary. This sort of thing has been seen before for like seagrass sand relationships. We're talking about much deeper sand patches here mostly, with boundaries onto other substrata. For cod, we have some we have a kind of relationship again, like higher growth rate with higher proportional coverage of algae. Again, not surprising given that we know that cod like these kind of algal dominated environments. But again, their highest growth rates are right on the boundary of patches of sand. So not exactly the same patterns as you get for max N from Sophie Elliott's work, or from probability of being observed, as we had for the previous work we showed from Natasha. So yeah, so growth results. Yes, the factors that drive growth are not exactly the same as those for max N and probability of presence. We think boundaries are coming out as being particularly important. There are likely to be interactions between cod and whiting on these. I should have said, sorry, we don't have data for haddock for this, just because there weren't enough caught in the year that the Marine Scotland guys were doing the trapping. So we only have cod and whiting for this part of the work. So the trap data show also, which I haven't shown the figures for this, but trap data show multiple settlements by whiting, but only one for cod, which is to be expected based on their spawning behaviour. And what was quite surprising with this is that the cod had mostly disappeared by the time of the second trapping in September, which is much earlier than expected. Although they can be very variable in terms of how long cod stay around in nursery areas for them to have basically disappeared by September was really surprising. So the question is really, is this a strategy, or is it evidence of a bottleneck leading to higher mortality? So what are the bottlenecks? So there are things that could be an issue, like how much of the habitat is available. It could be the extent of boundaries between seabed types could be really important, as it seems to be with like sand boundaries, for instance. And we think habitat quality, as indicated by benthic biodiversity, is important. But I think what is starting to appear in this work is that it's interaction with fish behaviour that creates the bottlenecks. So do they need something that's very rare, or relatively rare, or currently rare as a result of habitat degradation? Also, does the whole cohort need to be fitted into this juvenile habitat all at once? Obviously bad if you need to try and jam everything in. Everyone needs to be there at once. And how long do they need to be in it for? Obviously more of a bottleneck if you need your whole cohort to be sharing the same habitat for a long period. So these diagrams are a bit of an experiment. Hopefully they make sense to you. So what I have here are the three axes of the three different kind of dimensions. So the number of arrival waves, how long they need to stay in the juvenile habitat for, and then going this way, how restrictive the habitat need would be. We definitely don't want to be is over here in this kind of diagram, which would be that you need to stay in there for a long time. You have restricted habitat need. And you're all going to be there at once. So whiting, they arrive in a number of small waves. They're not trying to swamp predation by all arriving at once. They stay in a habitat for a relatively long time, but they have very unrestrictive habitat needs. So on this axis, they're very close to the origin. So that puts them here, a nice long way away from the danger zone over here. So they use something that's a habitat that's very common. They arrive in dribs and drabs, bit by bit over time, which means they're able to stay in the nursery area for quite a while. Cod then is very different from this because they're based on spawning aggregation. They're arriving in basically one big wave. They have the most restrictive habitat needs, based on a very narrow strip of habitat around the coast, at least in the West Coast population. It's very restrictive habitat need. So if they stay for any length of time, they're going to end up over here, which is this most dangerous area for bottlenecks. So what seems to be happening is they're basically not staying in the habitat very long. So they're very short occupancy, very restrictive habitat, and very small number of arrival waves. So they're here, and if they were to stay longer, they would be here, where it would be the maximum bottleneck danger. So what we don't know is whether or not this is an adaptation of this sort of group of cod on the west coast of Scotland, that they don't stay in their nursery habitat for a long time, or whether there is a bottleneck and it's basically just having very high juvenile mortality at this stage. So our conclusions on this are then that, so we have very clear differences for these three species. It's three important commercial species. Habitat quality, as indicated by benthic biodiversity, turns out to be really important for presence, but also for abundance. Drivers of growth are a bit different, and we don't know yet what is the most important thing, whether it's better just to have for a habitat, for its contribution just to hold loads and loads of fish, or whether it's a smaller number of fish is okay, as long as they all have like good growth rates and then go on to be successful as adults. And we don't know yet which of those is most important. Boundaries seem to be important, particularly for growth. And it could be that the availability of these boundary areas could be as an important bottleneck as the extent of the areas. But whether or not a bottleneck exists is an issue of nursery use strategy as much as it is the amount of nursery habitat. And if you have a single cohort and a requirement for rarer substratum type, that's going to make a bottleneck much more likely. And it seems to be much more likely for cod than it is for the other two species. And that might be related to the fact that cod is doing much worse than the other two species on the west coast of Scotland. So just real briefly, what we're trying to do next. So we've been looking at essential fish habitat stuff in Sundarbans mangrove forest. So we've been doing that work, and we're going to hopefully be continuing that with a PhD student or two soon. The program has expanded onto Loch Eriboll last year, led by Neil Burns, and hopefully to both Eriboll and Wester Ross this summer, which will give us a chance to see whether these patterns are general or not. And what we're trying to get to is to work out whether we can find better proxies for nursery area value that help us to prioritize these. In the coming highly protected marine area program, these criteria are still starting to be developed, but they do include what they describe as critical fish habitats as part of that. So there's a political commitment on this. But yeah, consideration of seascape and their ecosystem service value, I mean, that would be a good way to go, I think, in terms of the critical fish habitat bit, but probably also for drivers of biodiversity. Okay. So I've like cast of thousands to thank for this. If you're not on here, I'm very sorry, but a huge number of people have made amazing contributions to this work, and we've been funded by lots and lots of people to help us do it. So thank you very much, everyone, for that, and thank you all very much for listening. I hope you found it somewhat interesting, at least, and I look forward to hearing whether you have any questions.
Great, thank you very much, David. For anyone who's with us right now, if you have a question, please use the Q&A box at the bottom of the screen for David and I to see that. I can see a couple have already been popped in. And if there's anything that David has spoken about that you would like to recap and check out, then please look at the MASTS YouTube channel at the end of today. The recording of this talk and the Q&A session will be found there as well. So thank you very much, David. We've got a couple of questions already in the Q&A. And I actually had a question just relating to your screen that you shared just a couple of slides ago, which was about the term when it says critical fish habitat. Is that relating to a particular phase of the those three fishes' lives or anything? Like, is that just for nurseries or where adults are found as well? Like, when it says critical fish habitat, does that encapsulate the whole fish's lifespan?
Yeah, it can do. It's mostly refers to nursery and spawning habitat. So you may be aware of obviously the issues with the cod spawning habitat at the moment. Yeah, the spawning closure in the Clyde has obviously been politically quite difficult, should we say? The arguments about that at the moment. But yeah, spawning as well as nursery areas would definitely come under that. It could be other areas where they're just a higher density and a particular danger, say, of being over harvested in those areas. But those would be the main ones.
Okay, thank you. Right, we're going to jump into one of the first question that came in through the Q&A box, which states, so this was before you started talking about your bottleneck slides. This question came in, which asks, is it a direct link with diversity, or is diversity indirectly linked through the habitat types? Has this been accounted for in the analysis?
Yeah, so the habitat, the classification is in the analysis. Either classification in terms of Sophie Elliott's work, the first phase, or percentage cover of the different seabed types is included in the analysis already. So yeah, that's already accounted for. So it's over and above the effect of the substratum type or percentage cover. Yeah.
Mm-hmm. Okay, great. And then moving on to the second question, which was, are the results from Arran in terms of habitat preferences likely to apply on a wider scale for example, around the whole of Scotland?
Yeah, so I probably came to that like right at the end, in the we don't know for sure, which is why we're kind of trying to generalize this by having other study sites. So there's Loch Erebor, which it started on last year. Neil Burns, if he's here, maybe clarify, but I think a couple of hundred camera drops up there, 250 camera drops maybe in the first season. And those are currently being analysed, so we can see whether or not the patterns are the same there. And then due to move on to a third site during this summer. So yeah, so that is a key question. We don't know whether this is a general thing or not. We want to find out, and are moving on to try to work that out.
So Neil is in the chat, and he's just commented that it was 216 drops for you.
Perfect, thanks very much.
So our next question comes in and asks, how would the data differ if bait had not been used? Is that something you can actually comment on?
Yeah, a little bit. So Sophie did some very detailed work on the behaviour of the three focal species around the camera. So she looked at the exact positions of the fish relative to the bait, and the three species, as well as differing in all these other respects, they do definitely differ in terms of their like boldness and willingness to come to the camera and come to the bait. The cod generally don't really come to the bait that much. The whiting do, and haddock are sort of somewhere in between. So you would definitely get differences for whiting. You probably get relatively similar effects for cod, but in general, you always get higher numbers of animals seen at the camera if you have bait. That's the purpose of the bait. But the idea is to really kind of bring out the differences between areas, and we don't use the numbers seen as like an exact like a measure of population density. It's a proxy. We know it's max N is proportional to abundance, but the slope of the relationship between max N and abundance will differ between species. And so the bait affects that much more than others, depending on the species. So like I said at the start when I was talking about baited cameras, it's quite complicated to work out what the max N's actually mean. But they're a good proxy. It's been shown they are a good proxy for relative abundance, and that's what we use them for, to say this area has higher abundance than this other area.
Okay, thank you. So hopefully that answers that question. And actually the next question comes from the same attendee, which they're asking, how was the associated habitat determined by examining the otoliths?
From the GPS locations where the camera was dropped, and using the seabed map that had been that already existed at that point. So basically, we know where on the seabed the different seabed types are from the map, and we know the GPS location of the camera drops. So that's how we worked it out. We did suggest putting cameras on the traps just to see the seabed, but that didn't happen, unfortunately. And another time, that's definitely what we would do, to be absolutely certain. But that was how that was worked out.
All right, thank you. So the next question is from Alejandro, asking, is there enough substrate data available at the right resolution to be able to look at seascapes across the whole coastal area?
If you mean by whole coastal area, all of the west coast of Scotland, then probably not. And I don't think even in the kind of next phase of the work, we would definitely be able to answer that question. What I hope we'll do in due course is to be able to work out what the proxies are for seascape and seascape value, which we can kind of apply to a much wider area, and then and then test those things by basically making a prediction. Right, I reckon the seascape will be like this on this side of this island, and then go there and test it. We're not at that point yet. I think it's doable. I think it's definitely doable. I know how I would do it, but we haven't done it yet.
I see, future work ideas. We're going to move on to the question that followed that one, which was, are there lessons to be learned from the Magnuson-Stevens Act essential fish habitat concept that can be applied to critical fish habitats provision being developed here?
Yeah.
And I don't know what that act is personally.
Yeah, I think it's great. I mean, everything I've heard about it in terms of like it gets reviewed obviously periodically to see whether it's working. It seems like it seems like it's a very sensible way of doing things. I mean, as I understand it, maybe American people in the audience who would know better than me, but my understanding is that the fisheries management plans are allowed to vary quite a bit between states or between sort of NOAA fisheries areas, as long as they follow they meet these certain principles, right? And the protection of essential fish habitat is one of those. Because it's part of the law, it doesn't really give any opportunity for the authorities to neglect it, right? It has to be information has to be gathered about it, and they have to be able to show to their kind of stakeholders and people who might sue the government that they have taken this essential fish habitat into account, and as a result they know an enormous amount about essential fish habitat in a way that we haven't here yet. So it's not that we couldn't, it's just not had the same level of attention. But it's a requirement that they know about, that it's known in the US system. So therefore it is,
Okay, that might be something that I Google later because I've not heard of that act myself, so I might just check that out. So we're going to move on to David's question, which is, do you have any insight on diurnal patterns in abundance or use of the habitats?
Yeah, well, thanks very much for that. No, we don't. We have been restricted to doing stuff in daytime so far. I mean, most of our work has been, in sort of late summer. The fish sort of start arriving sort of late June, where it's kind of light pretty much the whole time. But no, this was a criticism we got right away from the very start of our first papers on this, is that we might be missing particularly the vital importance of seagrass, because all the fish might be going there and hiding in the seagrass at night, and we might be missing that. But no, we don't know. We don't know that. Again, it's clearly possible to do it. We'd need to be. It gets a bit more fiddly because we're obviously using lights in the daytime. You're obviously going to be affecting the fish somewhat. Using lights at night, obviously going to be affecting their behavior more. So we probably need to go to using a different light frequency rather than the standard white light that we use at the moment. So, it's definitely not impossible. It's a bit harder. One of the things I would really like to do is to track fish across these areas, so that we could look at individual movements that way, and that might be a very nice way of looking at diurnal movements. But the juvenile fish are pretty small, so we might be bumping up to the kind of limits of what's possible in terms of tag size. I'm not sure. And that's another one on my wish list of things to know.
Yeah, that'd be really interesting to see that track data. I'm sure there's someone out there who has the technology, someone in MASTS must have, be able to help out.
It's the smallest VR tag that you have, yeah.
Absolutely. So we have one last question in the Q&A. So if there's anyone who's still here and watching and you have a question, please just pop it in the Q&A chat. So this question that we have left in the Q&A says, do you have any comparisons between methods such as trawls versus traps versus BRUVs that would allow you to consider variable data sources and refine your abundance estimates to work out selectivity?
Yeah, so it's got done a bit in the deep sea. So Priad and Merritt did some stuff on this, looking at otter trawl data versus beta camera data in the deep sea. And there was a relationship, not a massively strong one, but there was a relationship there between catching trawls and the arrival time of the camera. So basically the sooner the sooner fish arrived at the camera, the more abundant they seem to also be in trawls in the same area. There's less of that been done in shallow temperate zone, but it's also much more complicated in shallow water because the currents are much more complicated. So in the deep sea, you have quite slow, predictable currents over large areas. So it's kind of easier to work out what the odor plume from the bait is doing. In shallow, like coastal environments, it's kind of a mess. There, the water flow is kind of going all over the place, and then if the current changes, then you can end up with this extremely kind of complicated kind of odor mess. And obviously, the longer the drop goes on for, the harder it is to make sense of that. We've had a couple of goes at working this out. So we did two projects on this. One of them was in Antarctica and the other one was in the Red Sea, where we did camera drops versus diver transects. And this is Kathy Dunlop's work. So right the way back from when we first started on all this. And she also did mathematical models of basically random searches by animals. And she basically populated those models with like different abundances, and then using the simulation, like how many fish would you see at the camera based on different odor plume characteristics and velocities and all that sort of thing. So yeah, our kind of conclusion to all of that stuff is that yes, MaxN is a decent proxy for the abundance you see by other methods, but it's complicated by things like if fish hide in holes, like moray eels or whatever, you don't see them in the transects, but you do see them at the cameras. And then you have other things that you'll see in the transects, but you then don't come to the cameras. So, we've probably done as much on this as anybody in the world, and it's still what we've ended up coming down to is it's the best thing to do is to use it as a measure of relative abundance, using the MaxN as a reasonable proxy for relative abundance. And from a practical point of view, that's the best way to go.
Great. Thank you very much, David, for answering all those questions. And that is the end of our questions. So that leaves me to say thank you very much, David, for being today's presenter. And thank you to all our audience for joining us today and submitting questions. And if you want to find out more, you can contact David directly and also check out the recording of this if this is something that you want to share with a colleague, or someone hasn't been able to join, you can just get them to check out the recording as well on the MASTS YouTube channel. So thank you very much, David.
No problem.